Hello everyone, my name is Yuxi and I'm delighted to be here today to present my study. I'm currently a graduate student at Nanjing Normal University, major in agriculture. My supervisors are Professor Ying Shaowu and Wang Tao. And the topic of my presentation is copper-induced intestinal inflammation response through oxidative stress, induced endoplasmic reticular stress in tachyfugal fasciitis. Next, I will present my study content from the following four aspects. Firstly, is the background of the study. T. fasciitis is a fish that mainly found in the coastal water of China, Korea, and Japan, and it has high economic value. And currently, the widespread use of copper has resulted in copper pollution in the aquatic environment and made T. fasciitis inevitably distributed by external copper exposure. And the intestine is like a crucial player in the digestive system, which can directly interact with the water environment and handle environmental stress. And having mental pollution in water can lead to an imbalance in the intestine immunity of fish. The endoplasmic reticular stress is a protective stress response in the cells and can activate three downstream unfolded protein response signaling pathway, the PERK-EIF2 alpha, the IRE1-XBP1, and ATF6 signaling pathway. Although many studies have shown that high copper concentrations can induce the production of reactive oxygen species in the organism and causing ERS and inflammation disease in the mammals. The relevant studies in fish have yet to be conducted. So in this study, we investigated the effects of ERS and its immediate UPR signaling pathway of copper-induced immunity in the intestine cells of T fasciators through in vivo and in vitro experiments. Hoping these results of this study can reveal the mechanisms underlying the copper toxicity in T fasciators and provide some basic information for the rational use of copper in the culture of T fasciators in some way. Secondly, is the research content. Here is a technological roadmap of this study. And this study was divided into two parts, in vivo and in vitro experiments. First are the in vivo experiments. T fasciators are treated with control 20 and 100 micrograms per liter copper groups for 28 days, and we analyzed the wet gain rate and copper accumulation levels of T fasciators, as well as the change of oxidative stress, ERS, and inflammation responses. And for the in vitro experiments, we cultured the primary intestinal cells of T fasciators and used the CCK8 assays to determine cell viability on the different concentrations of copper exposure. And in order to explore the relationship between oxidative stress, ERS, and inflammation responses under copper exposure, we added an ERS inhibitor for PBA and a RS inhibitor NAC to treat the cell for 24 hours for subsequent studies. Alright, let's moving on to the third part. I'd like to share the result and analysis of our study in three aspects. First is the in vivo experiments in weight gain rate, intestinal copper copper accumulations, tissue structural changes, and transcriptomic analysis in T fasciators under copper exposure. Now, here is the determination of wet gain rate and copper accumulations. As you can observe in Figure 1, there was no significant differences in weight gain rate between the control and 20 micrograms per liter copper groups. While there was a noticeable decrease in weight gain rate in the 100 micrograms per liter corpora groups and compared to the control groups. And additionally, the accumulation of copper in the intestinal of T fasciator increased with an increasing copper concentration. So we can infer that copper can inhibit the growth of T fasciators with intestinal copper accumulations. Next, let's look at the change in the intestinal tissue structure. From the picture, we can see that after 28 days of treatment, the intestine of the T fasciators in the control group showed normal structure. However, in the 20 and 100 micrograms per liter copper exposure group, different degrees of damage were evident in the intestine, including the inflammations. The damage increased with increasing copper concentrations. And then is a transcriptomics analysis in the 20 micrograms per liter copper exposure group. We found a total of 317 differentially expressed genes, with 207 upregulated genes and 110 downregulated genes. As for the 100 micrograms per liter copper exposed group, we identified a large set of 931 genes, including 444 upregulated genes and 487 downregulated genes. And the figure A represents a Venn diagram of DEGs. It shows that there were 118 common DEGs between the 20 and the 100 micrograms per liter copper exposed group. And the figure B represents a heat map of DEGs with the same expression trends in 118 common DEGs, and the star indicating that for ERS and for inflammation response related genes were significantly upregulated. And figure 5 shows the KEGG enrichment and analysis of the common DEGs, showing that 12 KGG pathways were significantly expressed. Among these pathways, these two signaling pathways are the most prominent. And the second aspect of the result is the effects of the copper exposure on ERS and the inflammation response in the intestine of T fasciators. And through the in vivo experiments, we investigated the intestinal oxidative stress enzyme activity. As shown in figure 6, the activities of SOD, CAT, and GSHPX and MDA increased significantly with increasing copper concentration, indicating that copper induced oxidative stress in the intestine of the T fasciators. And then the figure 7 illustrates the ultrastructural change of T fasciators intestinal. After exposure to copper, the nucleus was significantly deformed, and the endoplasmic reticulum was extremely damaged, suggesting that copper induced ERS in the intestine of in this fish. And figure 8 is the in vivo RT-PCR results of ERS-related genes and inflammation response-related genes after exposure to copper. The expression of ERS-related genes like GRP78, CRT, IER1 alpha, and XBP1 was significantly higher compared to the control group. In contrast, the expression of PRK, EF2 alpha, and ATF6 alpha gene did not show significant difference from the control group. This indicates that the copper exposure may activate the IRE1 alpha XBP1 pathway in T fasciators, and the copper exposure also lead to an increase in the expression of inflammation inflammation's response-related genes. These results were consistent with what we observed in the transcriptomic analysis. And the final aspect of our study's results shows that copper-induced intestinal oxidative stress triggers ERS and participates in regulating the inflammation's response in the intestine of T fasciitis, and the following are all the results from the in vitro experiments. And the figure 9 shows the effects of different copper concentrations on the cell viability after 24 hours of treatments. As you can see, cell viability significantly decreased with the increasing copper concentrations. The half lethal concentrations was approximately 30 micrograms per milliliter, and a 1/4 half lethal concentration was selected for subsequent experiments. And figure 10 illustrates the ultrastructural change of T fasciators, intestinal cells. And in the picture C is 7.5 micrograms per milliliter copper exposed group, and the endoplasmic reticulus was damaged, and the nucleus was several deformed. And in the picture D, with the addition of 10 millimolar 4 ppA, the damage was elevated. And the figure 11 shows the QRT-PCR results of ERS-related gene after additional of 4 ppA. The expression of ERS-related gene, including GRP718, GRP178 and CRT, ATF6 alpha, PRK, EIF2 alpha, IRE1 alpha, and XBP1 was significantly upregulated on the copper exposure compared to the control group. And this indicates that copper can activate the PRK, EIF2 alpha, IRE1, XBP1, and ATF6 alpha pathway in the intestinal cells of T fasciators. And after the addition of 4 ppA, the expression of all these genes decreased significantly. And the figure 12 is the QRT-PCR results of inflammation's response-related genes after the addition of 4 ppA. We can observe a similar pattern in the changes of the expression of inflammation-related genes. This suggests that copper induced an inflammation response through ERS in the intestinal cells of T fasciators. And in the figure in this figure shows the microscopical images of DCFH, DA fluorescence RS formations. The most pronounced fluorescence intensity was observed in the 7.5 micrograms per milliliter copper exposure group, and the addition of NAC promoted a significant decrease in the RS level. And from the figure 15, we can see 14, we can see that after the addition of NAC, the expression of all ERS-related genes was reduced compared to the copper-treated group, and this indicating that copper-induced ERS through ROS in the intestinal cells of T fascicle in this fish. And the same changing trend is also observed in the inflammation's response-related genes. So we saw that copper-induced inflammation response through ERS induced by oxidative stress in the intestinal cells of T fasciators. And finally, let's conclude our studies. Our study suggested that copper exposure significantly inhibits the growth of T fasciators and induces copper accumulation and structural damage in the intestine. Overall, through in vivo and in vitro experiments, we found that the copper exposure induces ERS and its mediate UPR signaling pathway through ROS, triggering an inflammation response in the intestine of T fasciator. And here is the innovations and the prospects. We hope that in the future we can investigate using gene editing to further study. And sincere thanks to the Professor Ying Shaowu and Wang Tao and all the friends in our lab for their help. And sincere thanks to all the experts and who participated in this lecture. And the relevant research content has been published in the Journal of Aquatic Toxicology. Thank you, guys. Bye. Thank you.